So, Profesor Kietsuda akan menerangkan lebih jauh tentang etilen yang kita sudah dengarkan waktu itu penjelasan dari Bu Titik ya. Nah, nanti mungkin akan diperdalam dan dipertegas, diperdalam lagi oleh Profesor Kietsuda. Nah, kemudian bagi mahasiswa pasca panen yang mendengarkan kuliah hari ini, sesudah ini nanti ada tugas yaitu membuat summary dari kuliah yang diberikan oleh Associate Professor Kietsuda Luengwilai. Okay, I think I already asked to my students, Dr. Kietsuda, that they need to draw a summary or to write in one page about your lectures so that they will understand better about the ethylene. Now, I think time is yours, Professor Kietsuda, and Pak Chandra will be your moderator, or if students asking some questions, he will be more than happy to translate if the students cannot speak in English. And yes, Pak Chandra, siap ya? Okay, I think time is yours, and good luck.
Okay, thank you. So I hope that by the end of the lecture, students can learn something new in this topic and ethylene in relation to ripening and senescent. Although the topic is in agricultural product, but I'm going to focus more on the horticultural product, vegetable, fruit, and flowers. Okay, ethylene is a plant hormone, one of the plant hormone, which is differ from other hormone in that they are very small molecule and they is in a gas in nature. These ethylene has the function in inducing ripening and senescence. The other special of this hormone is that they are also our plant can produce the ethylene, or tissue or plant tissue can produce the ethylene, which is different from anything else. Okay, and this ethylene. Okay, I think I can hear some background. Yeah, so the concentration that plant can be respond to the ethylene is very low. It's up to 0.1 ppm. So this is some background of the ethylene. If you're talking an ethylene, one thing that will be very common to mention, that is the triple response. This is the typical bioassay when people want to determine the effect of ethylene. So what is the triple response? The triple response is the response of the seedling, the bean seedling, under the dark condition. Usually you will see in the here in the picture, if you're growing this bean seedling under the dark condition, you will see the plant very thin and tall. What we call in this phenomenon, the technical term is etiolation. Okay, this is normal without ethylene. However, if the environment around this seedling had contained ethylene, what we can see, the stem of the seedling will be shorter as the concentration of ethylene increase. The seedling will be shorter. Okay, and another response is that the diameter of the stem will be thicker. Okay, and at the hypocotyl part of the seedling will become curved. So these three phenomenon, these three characteristics is the plant response to the ethylene. And then because they always have shown three characteristics, that's why we call triple with three and response, triple response. Okay, and this is what we use in the lab to determine the concentration of ethylene. If we don't have the expensive instrument, something like GC or gas chromatograph, we can use this response to determine how much of ethylene in the environment. Before go on to the ripening and senescence, I would like to introduce or talking about the ethylene biosynthesis. In order to understand ethylene better, we have to know how they synthesize and how they work. So the first thing we have to know how the ethylene synthesized in the plant. So this is a very simple diagram that I would like you to at least remember. This is important, very important. If you want to write the report, this is where you have to write it down. The ethylene biosynthesis starts with the methionine, which is the one of the essential amino acid. The methionine convert to S-adenosyl methionine, or SAM in short, by the enzyme SAM synthetase. This SAM converted into ACC, which is 1-aminocyclopropane carboxylic acid, by the enzyme ACC synthase. And this ACC converted into ethylene by the enzyme ACC oxidase, or the formal name is ethylene forming enzyme. This is very old name. You may not see this in the recent publication, but if you go back to the very old publication, you might see this name. And at the last step of the ACC to ethylene, this is the oxidation process. That means this enzyme will be characterized by the help of by the presence of the oxygen. Okay, this is very important, and we want every student at least had to know this pathway. And so since it is very important, I would like to remind you one more time. They start with the methionine amino acid. The chain to SAM, and the enzyme that involved in this step is SAM synthetase. SAM changed to ACC by the enzyme ACC synthase, and ACC changed to ethylene by ACC oxidase in the presence of oxygen. Okay, so although I show you the diagram, the basic diagram, but in plant, this pathway is happen in a cycle manner, which means that some part of the methionine molecule will be recycled to make a new methionine, and some part of methionine will change into ethylene. Okay. Yeah. So if we look at this molecule of methionine, they contain of 5 carbon atoms and sulfur. All right. And if we follow the pathway as I just mentioned in the earlier slide, the methionine will convert into SAM, right, by SAM synthetase. In here, at the step, methionine will bind with the ATP, okay, adenosine triphosphate. And this adenosine triphosphate eventually will be recycled to make a 4 carbon atom part of the methionine molecule. Another part of the methionine molecule will be recycled is sulfur. All right. If we follow this SAM, from SAM to ACC by ACC synthase enzyme, the enzyme will cleave at this position and release part of the ATP and sulfur into the cycle and make a new methionine molecule. The other 4 atoms of carbon will become ACC, okay, 1-aminocyclopropane 1 carboxylic acid, or ACC. And the first carbon atom of the methionine molecule will become carbon dioxide. The second carbon atom of this methionine molecule will become cyanide. And the third and the fourth molecule atom of carbon in methionine will become an ethylene. All right. So this is what happened in plant in terms of ethylene biosynthesis. And since this is the cycle, so we call this one methionine cycle or Yang's cycle. Yang is the name of the person who discovered this pathway. Okay. Again, to be simple pathway that at least all of students supposed to remember, methionine is a substrate changed to SAM by enzyme SAM synthetase. SAM convert to ACC by ACC synthase, and ACC convert to ethylene by ACC oxidase in the presence of oxygen. And here you can see the bigger picture, the overall picture. Since ethylene is the hormone that stimulate or promote the ripening and the senescence, which is the last stage of the plant life. So plants supposed to have a regulation to control the amount of ethylene. And if you see in this biosynthesis pathway, what we can see now, we have methionine, we have SAM, ACC, and ethylene. If we want to control the amount of ethylene, so the plant have three ways to control. They can control the amount of methionine, control amount of SAM, and control amount of ACC. For SAM is intermediate product, not only for ethylene, they're also a substrate for something else. For example, for polyamine biosynthesis. However, for ACC is not a substrate for any other metabolite in plant pathway, but they can convert it in the case of limit, if the plant want to limit the amount of ethylene, so they can convert this ACC into 1-malonyl ACC or MACC, or they can convert into gamma-glutamyl ACC or jasmonyl ACC. All of these compounds are not active. They don't have any function, known function in plant tissue. So basically, if the plant want to limit the amount of ethylene, they will convert, or what we can see in simple way, they can make ACC into inactive form, and they have multiple ways to convert into inactive form. All right. In plant, or especially for fruit, these ethylene had different regulation, and these regulation related to the climacteric and non-climacteric fruit. I hope by now you should know about climacteric and non-climacteric fruit, right? Janta, do students know the difference between climacteric and non-climacteric fruit?
Yes.
Okay.
The respiration.
Okay. So for ethylene regulation, they have two systems, what we call system 1 and system 2. In system 1, the plant had very low amount of ethylene. Okay. And somebody leave the microphone on. In system 1, the plant have very small amount of ethylene all the time. All right. And in system 1, all plant or tissue, no matter climacteric or non-climacteric, they all have system 1. Okay. For the system 2, this system 2 happen in the case of only climacteric fruit when they start to ripening. Okay, they start to ripen. It's already ripe. They will have the system 2. The difference between system 1 and system 2 is that for system 2, the amount of ethylene from system 1 will trigger the production of ethylene. So in this case, ethylene will become higher and higher as the fruit ripen. Okay, in here, again, system 1 is happen all the time of plant tissue, but system 2 is only happen in the climacteric fruit and only when it ripe. Okay, it's only will start to ripe. The another characteristic that differ between system 1 and system 2 is the amount of ethylene. For system 1, the ethylene concentration will be low at all time. For the system 2, the ethylene from the system 1 will trigger the production of ethylene. So eventually, they will have the higher concentration of ethylene in the system 2. All right. And if we combine those system 1 and system 2 and combine to the respiration pathway, what we can see will be something like in this picture. For this picture, if we follow that line, that will be the characteristic of non-climacteric fruit. In the non-climacteric fruit, the carbon dioxide concentration will be high after after pollination, when the when the flower pollinated and cell division happen, cell enlargement occur. So the concentration of carbon dioxide will be high at the beginning and then decreasing along the fruit development, and it will become lowest when the fruit mature. And after harvested the fruit will keep the low carbon dioxide concentration throughout the senescence process. Same thing with the ethylene. For the ethylene, the concentration, again, non-climacteric will only have system 1 ethylene regulation. So the ethylene concentration will be low all the time. For the climacteric fruit, at the beginning of fruit development until they start to ripen, the concentration of ethylene will be low. As soon as the fruit start to ripe, so the CO2 and the ethylene will increase. All right, follow the respiration. Okay, that is the ethylene biosynthesis. In order to get the response from ethylene, plant need this process. Okay, so we have ethylene biosynthesis, and we have to have, or plant have to have ethylene action. Okay, so if you only have biosynthesis, you're going to get the ethylene molecule. If the plant had to respond to ethylene, this ethylene molecule had to send a signal through the signal transduction process to get the signal into the gene, and gene will be transcript and translate into protein, into enzyme, and then plant start to get the response. This ethylene action is a little bit more complicated compared to other hormones. And the simple way to explain this ethylene action, if you look at this picture, this is the molecule of ethylene. So the molecule of ethylene had to bind to the receptor. Upon binding, they're going to send this receptor, going to send the signal downstream to other protein. And those other protein will transfer the signal into the gene eventually. All right. For ethylene, since it is a gas, after they bind to the receptor sensing the signal, And then this ethylene can release or can left out the receptor and bind to another receptor protein. Okay, and to become even more complicated in the cell, the ethylene, this is the ethylene biosynthesis. Again, you see methionine into SAM into ACC into ethylene. This happens in the cytosol of the cell. And ethylene is gas, so they can diffuse freely. They can dissolve in water and diffuse in the air, in the empty, in the space, intercellular space between the cell. If we want to get the response from ethylene, the ethylene had to bind to the receptor, right? So ethylene receptor is located in the membrane of endoplasmic reticulum, all right? And this receptor going to send the signal to the protein in the cytosol. The first protein receiving the signal and sent another signal into the second protein, EIN2, and this protein will receive signal from CTR and send the signal downstream to another protein inside the nucleus. All right, so we will see that this is a little bit complicated because they involve in too many these signal protein, signal transference protein. And this protein doesn't work in the simple way. Okay, I will try to explain slowly. The ATR, which is the ethylene receptor, going to promote or active the CTR protein. Upon receiving the signal, CTR will be inhibit or did not send any signal to EIN. All right. So meaning that in the presence of normal air without ethylene, if the air doesn't contain the ethylene, okay, ETR will become active. So ethylene receptor become active, sending the signal to CTR. And CTR is active, but CTR working negatively regulated EIN2. So if CTR active, they do not send any signal to EIN2. So EIN2 become inactive, meaning that they cannot send any signal downstream. So the gene in the nucleus will not active. In contrast, if the ethylene is present, the ethylene bind to the receptor in endoplasmic reticulum membrane. Upon binding, the ETR become inactive. If ETR inactive, they do not send any signal to CTR. And then CTR will become inactive too. And if CTR inactive, they don't have any negative regulated function. Ethylene, when we're talking about ethylene, I would like students to remember we have two important processes. We have ethylene production and have ethylene action. Ethylene production, you think of methionine, SAM, ACC, and ethylene. So ethylene production, eventually, the output we're going to get molecule of ethylene. In order to have those response, those molecules of ethylene had to bind to the receptor and send the signal downstream into the gene inside the nucleus. Those process we call ethylene action. All right, so they have two separate process going on, and this is very important because if we want to control the ethylene response, we can control both ethylene biosynthesis and ethylene action separately, right? Before going further, so different plants, different species produce different amounts of ethylene. Those, especially for non-climacteric and vegetable and flower, can produce very low amount of ethylene, things like cherry, citrus, grape, or cut flower or vegetable. For blueberry, cucumber, okra, the persimmon, pineapple, and raspberry, these produce low amount, about 1 to 1 microliter of ethylene per kilogram per hour at 70 degrees Celsius. For banana, honeydew, melon, mango, and tomato, these group of products can produce moderate amount of ethylene. Where apple, apricot, avocado, and papaya, peach, nectarine, these can produce high amount of ethylene. And most of the tropical fruit, especially for climatic tropical fruit, has very high amount of ethylene. This table is important because when we would like to store the fruit, one key important thing to keep in mind is that we do not support to mix the high the plant that
We lost your voice, Kietsuda.
Hilang.
okay, you lost my presentation again.
Yes.
Something with the recording progress, I guess.
Oh,
every
sorry.
time I got that message, yeah, my screen was gone. Okay. Okay, can you see my slide now?
Yes.
All right. So that is one thing to keep in mind. This is important table. It's good to know what fruit had high ethylene production and which fruit had low ethylene production in order to have a good storage condition. Okay. Apart of those species, other factors also affecting ethylene synthesis. For example, the first one, the variety. Here is an example of different durian cultivar, Chani, Kanyao, and Montong. Montong has very long shelf life, and Kanyao and Chani has very short shelf life. Okay, and as you may expect it, Montong has very low, not very low, but lower. Can you hear me now? Can you still hear me?
Yes.
Okay. But Chani and Kanyao has higher ethylene production. Okay, that is very simple. So the second factor is maturity stage. Again, this is also in the case of durian with 3 different different maturity at harvest. So the red line is the mature green. Okay, the blue line is immature, 10 days before mature. Okay, and green line is very immature, which is the fruit was harvested at 20 days before mature. As you can see now, for the mature green, the ethylene production start to increase after 2 days of harvest and peak at 4.5 days after harvest. However, if we harvest the fruit at younger stage or immature stage, the ethylene production will not increase until 4 days of after harvest. For the case of immature, 20 days before mature green, when the highest amount of ethylene, even lower than it's supposed to be if we harvest at mature green. Okay, so this is what going to be correlated with the quality. In the quality, if we harvest the immature fruit, you will not get a fully good quality. You may not get sweet taste. The smell will not be good as if you harvest at the mature green, for example. So this is the second factor, the maturity. The third factor that affecting ethylene production is temperature. As you may expect it, the higher temperature, the higher production of ethylene. This happens every fruit, every product. However, if the temperature is too high, for example, if the temperature go up beyond 35 degrees Celsius, then we start to see the reduction of ethylene production. Oxygen and carbon dioxide in the atmosphere also impact to the ethylene production. In the normal condition, in the air, they have about 20% oxygen, 0.04% of carbon dioxide. So if we lower the concentration of oxygen, for example, down to 4%, the ethylene production will be lower compared to the air condition. And that because if you remember, in the ethylene biosynthesis, oxygen is important, at least at the very last step when ACC converted into ethylene, at the ACC oxidase enzyme activity. Over there, they need oxygen. So if we have lower oxygen concentration, that's why the plant can have lower ethylene production. In contrast, if we get higher concentration of CO2, the ethylene production will be lower. Okay, for carbon monoxide has the effect mimic to ethylene function. For example, here, if you compare the treatment that contain only 4% oxygen and treatment of 4% oxygen plus 11% carbon monoxide, this apple will have higher ethylene production at this treatment where carbon monoxide is included. Okay. For ethylene treatment itself, the plant can produce ethylene, but if we apply ethylene externally, meaning if we fumigate the fruit with ethylene, what we're going to see, the effect in fruit will be different between non-climacteric and climacteric fruit. For non-climacteric fruit, in the control, the plant itself doesn't produce ethylene, okay, doesn't produce ethylene, and also didn't have a peak of respiration. However, if we apply ethylene for 1, 10, and 1,000 ppm, what you are going to see, you're going to see the increase of respiration. The higher concentration of ethylene will be the higher respiration rate. And if we take the ethylene out, the respiration will be dropped down at normal level. This is the response for non-climacteric fruit. For the climacteric fruit, in contrast, at the normal condition in the control, the fruit will have high respiration rate and will get to the highest point, to the peak, and after that will be decline again. If this fruit, climacteric fruit, was applied to the ethylene, 0.1, 1, 10, and 1,000 ppm, what happened here is that we are going to have the same amount of respiration rate. The respiration rate will not increase, but the peak, the day that you're going to see the peak of respiration will become shorter. For example, in control, this fruit had highest respiration about 10 days after storage, after harvest. But if we apply 0.1 ppm, the peak will happen faster at about 9 days. If we apply 1 ppm, the fruit will have this peak of respiration at day 7. And we apply 10 ppm, the peak will happen at about 5 days. And for 1000 ppm will be also for 4 days, for example. So if you can see in this picture, for non-climateric, higher concentration of ethylene, the fruit had higher respiration rate. And if you take the ethylene out, the respiration rate will drop down immediately after taking the ethylene out. For climateric fruit, the respiration rate will not increase upon ethylene treatment, but the day to see the peak of respiration will be shorter. The higher concentration of ethylene, the day to see the peak will become faster. All right. Okay. And the last factor affecting ethylene production is other hydrocarbon, things like propylene, carbon monoxide, which I already mentioned, and acetylene. However, these three compounds can have the effect similar to ethylene, but much lower threshold, meaning that in order to get response equivalent to 1 unit of ethylene, we need 100 units or 100 times of propylene, or almost 1,000 times concentration of carbon monoxide or calcium or acetylene in order to get the response only 1 unit of ethylene. All right. So in short, these or other hydrocarbon can have the response similar to what plant can respond to ethylene, but we have to provide higher concentration. Oh, a very last factor is stress. If the plant gets stressed from biotic or abiotic stress, the fruit will have higher ethylene production. Okay, in here, it's a case of tomato. If we drop the tomato 1 time, 4 times, and 8 times, the higher of number of dropping, the higher ethylene production will happen. Okay, and the last section of my talk, I'm going to show the student about the effect of ethylene on food quality or food ripening. First of all, ethylene will stimulate the ripening process. For example, the loss of chlorophyll and the softening. And also ethylene will stimulate or promote the sprouting of potato. However, in sprouting of tuber, this is a little bit complicated. If you can see here in control, in order to germinate the tomato to germinate 50%, for control it needs 30 days. If this potato is applied to ethylene 2.2 ppm, in order to get 50% sprouting, it needs only about 25 days. And higher concentration of ethylene.
Ini pagi-pagi ngetop gue.
Do the student ask question? Or they're just talking?
Not yet.
Okay. And higher concentration of ethylene, 0.2, 0.02, and 20 ppm. You will see that the tuber of the potato can germinate faster, all right? But when the concentration is higher than 20 ppm, the sprouting rate will again become lower. So with these results, I can conclude that the low concentration of ethylene promotes sprouting. 0.02 to 20 ppm promote sprouting, but high ethylene to 200 ppm, for example, this will be delay the sprouting of the tuber. Ethylene also promote the senescence of flower. In flower, the senescent characteristic can in orchid could be seen as venation or water soaking and duping of this petal. Here you can see only 0.4 ppm for 1 day can induce the senescent symptom. That means this flower is very sensitive to ethylene. Remember, if we want to prolong the shelf life of this flower, we're not supposed to keep this flower next to the fruit that produce a lot of ethylene, sapote, durian. Do not put these two fruit next to this flower, otherwise the flower will become senescent. In carnation, if the plant contain if the environment around the carnation flower contain high ethylene, the carnation will not bloom but go straight to senescent. This is called sleepiness. In longkong, ethylene will promote the abscission, which is going to reduce the quality of longkong, and also ethylene accelerates the softening. In the case of kiwi fruit, the higher concentration of ethylene will get lower firmness value. In watermelon, what we can have if the ethylene is around is this what we call maceration. You can see they have these bad looking and very soft texture at the pulp of watermelon around the seed, near the seed, and this happened because of ethylene. All right, ethylene also promote some pathogen infection. This is because pathogen itself has respond to the ethylene, or because ethylene promote the senescence of the fruit. And as you may imagine, if the fruit becomes senescent, the cell wall and everything else will be will be low. So that's why the plant doesn't have the defense mechanism enough to protect themselves from the pathogen. Another effect of ethylene in the carrot, the ethylene promote the bitter tasting compound in carrot. This thing happen in the storage or in the refrigerator if you keep the carrot for too long. And if you have apple nearby, those apple might produce ethylene, and you can taste these carrot will become bitter. And that's because those carrots, upon exposed to ethylene, they generate the isocoumarine compound. And this isocoumarine gives the bitter taste. In asparagus, ethylene will promote the peroxidase, which is an enzyme that can this is the important enzyme for lignin synthesis. So if the asparagus is exposed to ethylene, it will be toughening. And the last section, I have about 10 minutes. I'm going to talk about the source of ethylene. I already mentioned that in nature, all tissue or plant can produce ethylene. And also all microorganisms, ripening fruit, damaged fruit, or decayed fruit can produce ethylene. In the man-made source of ethylene, all of burning material, even though not only from gas, from oil, automobile, ballast, smoke, or from rubber, cigarette, or fire, or pollution, all of these things can produce ethylene. So ethylene is all around. So the plant, you have to keep the plant very safely from this because ethylene is all around. So in order to overcoming ethylene effect, we have 5 strategies. First one, avoidance. The second one, removal. And the third one, inhibit production. And the fourth, inhibit action. And the last one, which I may not have time to talk about, is the genetic engineering to modify the plant that does not contain the gene that respond to the ethylene. For the avoidance, what we can do, so we know that automobile smoke from the car can generate ethylene. So in order to avoid those ethylene, they recommended to use electric forklift or electric car in the packing house, and also avoid all pollution. Do not allow the co-worker to smoke in the packing house, and also do not store the product that produce a lot of ethylene with those products that are very sensitive to ethylene. For example, do not keep apple with the flour. Do not mix those 2 products together. Otherwise, the flour will be senescent faster. The second strategy to remove ethylene, what we can do in this case, we can do ventilation. For example, open the storage room to vent the ethylene inside the room out to the atmosphere, or using some ethylene absorber, things like potassium permanganate or activated charcoal, or use ozone or low pressure system. For potassium permanganate, you can see this one, we use this one to wash our vegetable in the kitchen. Potassium permanganate will oxidize ethylene to make manganese dioxide, potassium hydroxide, and CO2 and water. This very common in the packing house and in export market. Ozone also oxidize ethylene. The way to make ozone, we use UV radiation at 185 nanometer, and this UV lamp, at this wavelength will entionize oxygen and make this oxygen to become ozone. And we release this ozone into the storage room to oxidize ethylene and make carbon dioxide and oxygen. And remaining ozone will have to be get it off before we open the storage room. To get it off this extra ozone, we change the UV wavelength from 185 into 254 nanometer. At those wavelengths will turn ozone into oxygen, and then we can ventilate the air from the storage to outside. Using low pressure temperature storage, the idea is that if we lower the atmosphere, everything in the air will be lower. For example, at normal atmosphere 1 atm, we have 1.1 ppm. If we lower the pressure half to 0.5 atm, the concentration of ethylene in that atmosphere will become half too. It's only 0.05 ppm. That also works well to get it off the ethylene. However, in this strategy, we need to build a very strong storage room to tolerate this low pressure. So the third strategy to overcome ethylene effect is to inhibit ethylene biosynthesis. To inhibit ethylene biosynthesis, we have to inhibit enzyme in the pathway, which is ACC synthase and ACC oxidase. To inhibit ACC synthase, we can use a compound called AOA or AVG. To inhibit ACC oxidase, we can use carbon and anaerobic condition or high temperature. However, keep in mind in this side of the pathway, they will be promote ethylene biosynthesis, ethylene from outside, from ethylene from fruit ripening, ethylene from flower senescence, high oxygen, wounding, chilling, drought, or flooding stress. Those conditions will promote ACC synthase and eventually will promote ethylene production. All right, and ethylene and fruit ripening not only promote ACC synthase, but they also promote ACC oxidase. Okay, and the fourth strategy to overcome ethylene response is to inhibit ethylene action. In order to inhibit ethylene action, we can use low temperature storage. We can use a silver in terms of silver nitrate or silver thiosulfate. But silver is toxic, so they're not allowed to use for fruit and vegetable, but they use a lot in flower. Or use NBD, norbornadiene, but this one we did not use this compound anymore because they has very bad smell, and we have to have high concentration of the compound. But we use a lot these days to inhibit ethylene action by using this 1-MCP, 1-methylcyclopropene. And the last strategy is using low oxygen or high carbon dioxide in the pathway. Okay, this is the example where we put silver. Again, if you remember, silver is a compound that inhibit ethylene action. So if we put silver nitrate with the sucrose, we can get longer shelf life, vast life of flower. Ethanol also inhibit ethylene action. If we put ethanol and sucrose and sugar, we can prolong the shelf life of this rose. 1-MCP also inhibit ethylene action. If we apply 1-MCP, we can prolong the color change of this mangosteen. 1-MCP is the compound that inhibit ethylene action. I already introduced this figure when molecule of ethylene bind to receptor and send the signal, and then ethylene can go out to bind another receptor. But 1-MCP with the structure of 1-MCP, they mimics the structure of ethylene. So they can bind, they not bind perfectly, but enough to block the real ethylene to come to bind to the receptor. And upon binding, they bind forever. So in order to get a response to ethylene, plant need to build or to generate a new receptor. Okay, that's why for 1-MCP treatment, the plant can delay ripening, but eventually, if the plant can generate or can produce a new receptor, and if we apply ethylene, the plant still be able to ripen after 1-MCP treatment. Very last for my talk, if we would like to ripen our fruit, we have so many sources of ethylene. The first one is the ethylene gas from the tank. So we can buy this tank of ethylene. However, ethylene is exposure compound. So we have to have a very nice regulator to regulate this concentration and pressure of the ethylene. We also get ethylene from ethylene gas generator. And this idea, they boil the ethanol up to 350 degrees Celsius with aluminum oxide to get ethylene. And this also exposure product. Then we have very be careful using of this one. We can get ethylene from this ethyphon. The idea from this one, if they are in the bottle like this, the pH of this bottle is acidic with a strong acidic. Ethylene will be dissolved in water. However, if the pH above pH 5, the ethylene can be liberated from this solution. So since ethylene is a gas, so we don't really need to dip the whole fruit. We can dip only on the top, and the whole fruit will get ethylene gas eventually. Okay. Also, we can get acetylene and ethylene from calcium carbide. The idea of this calcium carbide will work when we apply a little bit of water, and then this water and calcium carbide together make acetylene, and about 2% of ethylene. Acetylene, if you remember from the previous slide, it mimics ethylene action. However, we need 1,000 times concentration of acetylene to be efficient as 1 time of ethylene, right? And this is very simple comparison between using ethyphon, calcium carbide, and ethylene gas to ripe banana. For banana that doesn't have a ripening treatment, the fruit will be green. But with this ethylene treatment, with different sauce, they can ripe faster and more uniform. And important thing to remember from ripening is that we need not too low or too high temperature, but we need very high humidity. And also we need to keep carbon dioxide lower than 1% in the case of mango. Otherwise, CO2 can inhibit the ripening, meaning that if you did not get rid of the carbon dioxide, although you treat with ethylene, but your product will not ripen. Okay, this is very important. Not too low or too high temperature, very high humidity, proper concentration of ethylene depend on how fast you want your fruit to become ripened, and had to have the treatment that get rid of those excess CO2. And very basic, or you can use other fruits, things like the fruit that produce high ethylene to ripen your fruit too. Okay, so with that, I would like to end my talk, and I will be more happy if students have a question.
Okay. Any student have a question for Dr. Kietsuda?
I don't know if they...
Anak-anak silakan bertanya, mahasiswa boleh bertanya. Kalau masih belum bisa pakai bahasa Inggris, pakai bahasa Indonesia juga nggak masalah, atau kemudian dicampur, nanti Pak Chandra akan menerjemahkan, atau saya juga menerjemahkan. Itu ada yang raise hand, Pak Chandra. There is one raise hand, Abimanyu. Pak Chandra.
Silakan, Mas Abimanyu.
Yes, thank you for the chance to ask for some question. Before that, yeah. Saudi Hub, Nano Hub. I want to ask how the ethylene being extracted and become a product. Jadi mungkin bisa minta tolong translatekan, maksudnya kayak gimana dari ethylene dari tanaman itu tuh bisa diekstrak dan dijadikan produk gitu. Terima kasih.
Okay.
Thank you.
So the question is, can we extract the ethylene from the plant to some like new product or some like new ethylene? Extract it and keep it like in the ethylene.
I I know that point. It's very interesting. So ethylene is a gas. Remember, it's not like a carotenoid or vitamin or something like that. And it's not good for human because we don't have receptor. We don't, and if you want to extract that one for ripen the fruit, right, or for the plant itself, since it is a gas, although you can extract, but it's difficult to keep those gas in the consent and they have other source of ethylene which is very cheap. It's from petroleum extraction. You know, those petroleum oil has byproduct as ethylene. It's very cheap. It's a waste of product from their process. So we can extract, I think, because we can measure ethylene, so we can extract the ethylene, but it's not worth it to keep or to sell this as a product for, you know, ripened fruit or something like that. Is that what they want? They question, and I get it right?
Terjawab nggak Mas Abimanyu?
Yes, for sure. Thanks for the answer.
Yeah, I think this is very good good idea, good question, because you know, if imagine if this ethylene is carotenoid, for example, and they ask, can we extract this one from the fruit and make more money? Yeah, that's a very good idea. However, for ethylene, other process is available already and it's very cheap already. It may not be worth it to invest on this one.
Yeah.
Very good one. I never get this question.
Thank you.
Very good thought.
Any question again for any student?
It's about lunchtime, I guess. The student probably get hungry.
Okay.
Ayo silakan, kalau ada yang bertanya, ini nanti kalian kan tugasnya membuat summary ya, jadi silakan.
It's a good talk actually, like very complete one. So among our participant is we have our president of the university, actually, Prof Gunawan.
Oh, good afternoon.
Yes, it's very interesting, Professor Kietsuda. So I'm very glad to join your lecture. And at the moment, we have also a project of using one MCP to prolong the shelf life of a banana and also the Salaka edulis or snake fruit. So I think your lecture gave me more insight about how to inhibit the ethylene to prolong the shelf life. That's very good, and I'm glad that I can see again the Yang cycle. Thank you.
Okay.
I used to have that Yang cycle, but then I don't know, I lost it somewhere, and then I tried to dig some of the books and also the journals to get the Yang cycle, but it's yeah, I couldn't find it, so I'm happy that I can find it. Thank you so much.
If you want the original version, I can send it to you too.
Okay, thank you. It will be great because I need to write a paper about the horticulture product, and I need the Yang cycle to explain more about some inhibitors of ethylene. Thank you so much. It will be great.
Okay. And for banana too, my colleague found that if you dip it into warm water, it depends on the cultivar and depends on the pre-harvest factor, but with warm water, it can prolong the shelf life, the inhibit ripening. Okay.
Oh, okay. So so it means that temperature in that case has a role in protect. Yes.
So the initial idea is that they found that the banana harvested during summer season,
Yeah.
it never ripened as it's supposed to be compared to other season, and that's why they think, oh, probably hot weather during summer season will play a role. And then they start to do the experiment where they dip into 40 degrees Celsius, 45 degrees Celsius.
45 degrees Celsius, yes.
Yeah, for one up to 1 to 5 minutes, depend on the size of the banana, depend on the cultivar, and with that the banana can be prolong the green color and the texture and the ripening. But they're not consistent yet, so they don't know. It had to be the pre-harvest factor.
Yes.
But they cannot confirm or conclude at the moment how to be precise how hot or how long to do that. Yeah, but that is another option that is going on in banana project in our country. Yeah.
Yes. Okay, that will be interesting, Pak Chandra, for the next research project. Yeah. Yes, because in banana, if we succeed to prolong the shelf life using the hot water, it will be cheaper.
Yeah, and we don't have to worry about the residual chemical too.
Yes, that's true. Yeah. Okay, that's very good.
Okay.
Yeah. So the students, mungkin ada lagi yang bertanya? Ayo silahkan bertanya satu atau dua pertanyaan yang berkaitan dengan ethylene. Sudah jelas semua ya. Silakan kalau ada yang masih bertanya. Kalau disuruh bertanya pada diam semua.
Itu aja.
Somebody raise their hand.
Monggo Mas Septian.
Okay, Septian.
So you give extra credit, right? If somebody asks question.
Yes.
Nggak bisa pakai bahasa Inggris, pakai bahasa nggak apa-apa ya Pak ya? Bahasa.
Boleh.
Tadi kan yang di awal-awal itu ada kayak semakin banyak konsentrasinya itu kayak tumbuhannya kerdil loh, cambahnya gitu. Terus sama ini sih apa sih kaitannya sama ethylene sama karbon dioksida sama oksigen itu gimana? Apakah semakin tinggi konsentrasinya itu buahnya akan semakin tua atau malah sebaliknya gitu? Itu sih tadi antara konsentrasi oksigen sama karbon dioksida itu apakah semakin nanti tinggi itu malah tua atau semakin rendah itu malah sebaliknya gitu, intinya itu.
First question is, can you explain again the correlation between ethylene with the first picture that you saw, the seedling?
The triple
response, right?
Yes, triple response.
And then the second one is the correlation of ethylene with the concentration of oxygen and carbon dioxide.
Okay.
So like the higher of oxygen, what the effect of to the ethylene.
Okay. So the first one, triple response, right? So ethylene inhibit the ethylation. If you know ethylation is the plant growth under dark condition. Imagine if you grow the plant under the dark condition, you get the very slim seedling, right? But if that environment contain ethylene, you will not see ethylation anymore. The higher concentration of ethylene, the plant will become shorter, okay, and become thicker, and at the hypocotyl will curve. Okay, and with this, since they have the correlation, the higher concentration, the shorter of the stem and the thicker of the stem, the shorter and thicker of the stem, and usually it has a not linear correlation, but it has some correlation. So with that response to ethylene, and the response depending on the concentration of ethylene, we can use, you can measure the height of the stem of the seedling to determine how much ethylene is in the atmosphere. Probably I had to explain this. And the second one, for the higher concentration of ethylene, oxygen is important for ethylene biosynthesis, especially at the very last step when ACC turns to ethylene. At that one, the enzyme ACC oxidase needs oxygen to stimulate this process. All right, so that's why if we have lower concentration of oxygen, we will get lower ethylene production. And opposite will be happen with this carbon dioxide. If you have higher carbon dioxide concentration, in proportion will be lower oxygen, right? And CO2 also being act as a competitive to the oxygen to bind to the enzyme. And also they have some researcher believe that CO2 also competitive to ethylene to bind to the receptor. However, we don't have a strong confidence to confirm the second property in terms of ethylene action, but for sure for ethylene biosynthesis, high CO2 will be competitive to oxygen to promote the ACC oxidase. Okay, in conclusion, lower oxygen, higher carbon dioxide will inhibit ethylene biosynthesis, especially at the ACC oxidase step. Okay.
Sudah jelas Mas Septian, apa perlu dijelaskan?
Perlu soalnya agak susah nangkepnya cuma.
Baik, saya coba ya. Jadi yang pertama yang waktu di sidering tadi, jadi kondisinya kalau di gelap itu kan, kalau di penumbuhan biji benih ya, itu nanti kan akan memanjang ya karena gelap. Etiolasi kalau di Indonesia. Nah, dengan kondisi itu ketika ditambahkan etilen maka akan merubah bentuknya. Jadi misalnya lebih thicker, lebih gemuk ya. Tapi tidak terlalu panjang. Semakin tinggi konsentrasi etilennya semakin pendek, semakin semakin nggak memanjang lagi. Jadi dan semakin gemuk ya misalnya gitu. Itu. Jadi kita bisa menggunakan itu sebagai cara untuk mengukur etilen di dalam ruangan gelap. Gitu Mas Septian, yang pertama. Yang kedua tadi mungkin langsung di konsentrasinya, langsung di kesimpulannya adalah semakin rendah oksigen dan semakin tinggi karbon dioksida akan menghambat sintesis dari etilen. Mungkin itu. Kalau sebaliknya misalnya tingginya oksigen, karena oksigen berfungsi pada proses sintesis ini ya, etilen. Jadi kalau semakin tinggi oksigennya maka akan semakin banyak pula etilen yang disintesis.
Saya tambahkan ya Pak Chandra ya. Saya tambahkan sedikit ya Pak Chandra. Jadi Septian, terima kasih pertanyaannya. Jadi tadi Profesor Kietsuda dan Pak Chandra sudah menerangkannya tentang etiolasi. Nah, mungkin kalian bertanya terus fungsinya apa itu untuk memahami itu. Nah, karena begini, kita untuk mengkuantifikasi atau menghitung etilen itu sangat sulit karena tadi sudah diterangkan Profesor Kietsuda ya, bahwa etilen itu berupa gas sehingga kuantifikasinya agak sulit, kuantifikasi, ekstraksi maupun penyimpanannya. Nah, salah satu cara Cara yang paling valid adalah menggunakan gas kromatografi atau GC, ya. Tapi itu kan mahal, ekspensif. Nah, sehingga kita bisa melakukan pendekatan melalui tadi yang namanya etiolasi. Jadi kalau pada 0,1 ppm kondisi kecambahnya seperti ini, pada konsentrasi sekian ppm kecambahnya seperti ini. Nah, sehingga dengan menggunakan paling tidak tolak ukur itu kita bisa memprediksi berapa banyak konsentrasi ppm yang ada di dalam pengemasan itu. Itu sebagai satu pendekatan saja. Kemudian tadi yang dikatakan tadi tentang apa konsentrasi oksigen ya dan sebagainya, itu perlu kita pahami. Tadi Profesor Kietsuda sudah menyampaikan tentang sintesis atau biosintesis dari etilen. Jadi ada di yang cycle itu ya, dari metionin ke SEM kemudian ke ACC. Nah, itu kan ada ada siklus-siklusnya. Nah, sehingga kita menghambat sintesis etilen itu dari siklus-siklus seperti itu. Seperti kita hambat ACC oksidase supaya nanti ACC itu tidak berubah menjadi etilen dan sebagainya. Jadi itu merupakan satu proses ya, bagaimana inhibitor berperan di dalam menghambat atau blocking sintesis etilen, termasuk juga nanti kaitannya itu dengan oksigen ataupun dengan karbon dioksida ataupun dengan karbon monoksida CO. Itu nanti kalian lihat materinya Prof Kietsuda, di situ ada bagaimana pengaruhnya. Baik, matur nuwun Pak Chandra. Sorry, Profesor Kietsuda, I explain the student using bahasa Indonesia so they can understand better. Yeah.
No problem.
I think so. We are over limited. The time is over. So
Okay.
thank you very much for today, Achan Kietsuda or Dr. Kietsuda for coming for join our lecture today. So thank you very much for your can I say your time to give
My
us
pleasure.
to give us especially our students to give us like enlightening lecture. Thank you very much.
Hopefully they can learn something new and interesting and yeah. And so I give slide. You got my slide already, right?
Okay. I sent it already.
Yes.
Okay.
Thank you so much, Professor Kietsuda. It's been very wonderful, and I believe that my students can now understand better about the ethylene. And I asked them to write down a summary about 1 page of your lecture. So I pushed them to study more about the cyclus and everything. I know if I ask them to study about the cyclus, they are reluctant, but by hearing your lecture, I'm sure that they will understand much better.
Okay.
Thank you very much. I really appreciate that.
Okay, you're welcome. Okay, have a nice day. Bye bye then.
Yes, you
Bye.
too. Have a nice day. Take care.
Bye bye.
Thanks. Sawadee-krap. Baik Bu, mungkin itu.
Ya, silakan. Nanti untuk yang mahasiswa pasca panen, jangan lupa bikin summary-nya. Kemudian nanti di My Class akan diberikan wadahnya. Kalian bisa upload untuk assignment-nya. Assignment-nya atau tugasnya adalah membuat summary dari kuliah yang diberikan Associate Professor Kietsuda pada siang hari ini. Kemudian tadi sudah direkam ya Pak Chandra? Sempat direkam tadi. Nah, ya nanti bisa diedit rekamannya ya, mungkin tidak the whole story, atau kalau cukup nanti langsung diupload saja di My Class sehingga nanti anak-anak bisa memahami